All right. So we're continuing with this idea of diffusion, which is really just kind of imagining we could zoom down to the level of molecules and atoms and watch how they move. And diffusion is describing a particular kind of movement that is really, I mean, if you understand diffusion, you understand most of biology, to be honest with you. So we're going to take a look at that. Hopefully you're building your own mental model, kind of the way you picture and understand diffusion. There's lots of great externalized models out there. So I've got this one running right now. This is a model of just some water. If we could zoom down and see what water molecules are doing when they're sitting there in the cup on your desk, this is kind of how they're behaving. Now, it just looks like kind of a jumble when they're like this. So the cool thing about this simulation is we can just pick one and watch what it does and put a little tracer on it to see what determines its movement. So at this point, you've played around with a couple of PhET simulations about how things diffuse, how molecules move. This is just kind of driving home. This is a pretty simple simulation. If we track one molecule, we can see that what tends to be the rule here, and you can see that this molecule makes straight runs from time to time. So that's really how molecules are moving most of the time. If you don't mess with them, they're just going to cruise along in a straight line. And if they change their direction, if they change their path, it's because something interfered with them. So you can see if we trace this little purple dotted line here, we can see that molecule is taking kind of a wacky path around. It's not able to really move in a straight line for very long. It makes little runs like that, but it's always getting bumped into by other molecules because they're all doing the same thing. They're all moving as well. And they're all continuing on a straight line until something bumps into them. And mostly they bump into each other in this simulation. So we can, slow this down and go into super slow motion. And you can see that red molecule, the one that we're tracing, it sometimes actually sort of holds still depending on how it gets knocked into. So it has times where it speeds up, times where it slows down, times where it changes direction. But that's always happening because it hit something or something hit it. So it's really, we want to think about the way molecules are moving like you're in bumper cars at an amusement park, right? You can go in a straight line, but then somebody slams into you from the side and it knocks you to the left, or you get knocked to the right, or you have a head-on collision, you get knocked backward. So that's always happening. So we go back to regular speed. We're going to reset this so we're not tracing that molecule. Now, we've also got, in the mental model now, you've hopefully played around with temperature. So you can see if I crank up the temperature, how the molecules behave differently. If I make the water very cold, the molecules, they're still moving. So even when water is very, very cold, the molecules are still moving. But as we mess with the temperature, we can speed them up and slow them down. Now, I have this option to add dye, so I can drop a little, like, you can imagine I'm going to add food coloring to the water. So if I add food coloring, we can see what happens to the food coloring over time. The food coloring is made out of molecules too. They follow the same rules. Each molecule is in motion. It's going to continue in a straight line until something messes with it, until something knocks it backwards or knocks it off its path or sends it left or right or it hits the walls of the container. But ultimately what we see is this consistent tendency, because everything's in motion and it's just randomly moving around and it goes in a straight line until you knock into it and it's not going in a straight line anymore. Eventually we see the dye spreads to every corner of the container and nothing stops moving at that point. But at that point we've reached what we call equilibrium. Everything's kind of equally spread out everywhere. It's still in motion, but we're no longer seeing that kind of spreading out. So we see that when I add the dye, even if I try to start the dye way over here in the right-hand corner, if we watch it, and this is a little bit cooler, so things are a little bit slower, the dye won't stay in one corner. And that's because everything's in motion. The only way you could get dye to stay in one corner in this simulation is if you just stopped the motion. And that never happens in reality. Molecules are always moving. So because they're in random motion, they move in a straight line until something bumps into them, they're going to spread out everywhere. And that's what we're looking at when we're looking at equilibrium. Is everything just kind of spread out everywhere. So when we look at, where did I want to go? I wanted to go to this. So we have a mental model of diffusion that says molecules are always in motion, they're just moving randomly, they go in a straight line until something pushes them off that straight path, so they're always crashing into each other. But if you give them enough time, they'll spread out to fill whatever space they're in, right? So they'll kind of spread out equally everywhere. So we have this model of, I'm trying to move this so you can see it. We've got a model here, that's about as far as I can go, of actual molecules. So we've been treating molecules like they're just little BBs, little spheres, and that's a perfectly fine way to think of your mental model for the most part. We can get more sophisticated and say, okay, some of those particles are water, some of them are oxygen, some of them are carbon dioxide. They can be all kinds of different things that a living thing has in its body. And we've been treating membranes as sort of just a simple wall or just a barrier that will keep things in or out. And in this scenario that I'm showing you with this very messy picture, I know it's very busy, it's not aesthetically pleasing, but we've got a little legend over here. This is a cut off a little bit, but I'm going to go to my drawing tool. We've got oxygen, drawing. Here's our oxygen, right? So anytime you see that kind of double BB, that's an O2, an oxygen molecule. And so you can see them in the picture. And then we've got a couple other things up here. So we've got, these are sodium ions, and these are potassium, which is weird because it's like, why would K be for potassium? But it is. And why would Na be for sodium? Anyway, Latin, Greek, don't ask too many questions right now. So these little diamonds here represent potassium, and these little round spheres represent sodium. So I'm just going to put a note so that you know sodium and potassium. Okay, so we've got a bunch of spheres up here. So we've got a lot of sodium ions on that top side of the membrane. Excuse me. If you look down at the bottom of the membrane, so you can kind of imagine that this is inside the cell, and this is outside the cell. Ooh. Okay, so we're imagining a freeze frame of, this is a living cell, and we've got a membrane running through. So we're looking at both the outside of the cell and the inside of the cell. The membrane is what separates the outside of the cell from the inside of the cell. And we know now that membranes can have these kind of tubes through them, these channels. Some channels can open and close, other channels are open all the time. We're imagining a scenario just before the channels open. So we're going to imagine these channels look like they're open right now. We're going to pretend they're closed at the moment. All right, so we've got sodium channels here, this kind of hot dog bun looking one, and potassium channels here. Because as you've seen from previous simulations, channels are specific. So just a channel through a membrane doesn't let just everything through. They're specific. They're shaped a certain way. They're built a certain way that only certain things can get through. So the hot dog bun looking one only lets sodium ions get through. The potassium ion one, the purple one, only lets potassium ions get through. So we have to remember our code over here for which is which. So at the moment that we're imagining right now, the channels are closed, and that means that those substances, the sodium and the potassium, can't pass through this membrane. They need to travel through those channels, and if the channels are closed, they have no way to get there. So we're imagining in our diffusion model that all these little sodium ions are moving around in straight lines until they bounce off of each other. Oh, and they're bouncing off the membrane. So they can't get through the membrane. Once where their channel opens, they'll be able to move. Now we know from our mental model of diffusion that if you open a channel for them, they're not going to stay put. They're going to keep doing their random motion, crashing into each other, and just randomly, some of them will kind of bob through that channel. So the scenario we're seeing up here is we have a lot of these sodium ions outside the cell. Now, if we compare that to inside the cell, all right, I see 1 here, 2, 3, 4, 5, 6. So there's not very many sodium ions inside the cell. Now, this is what we call a concentration gradient. Concentration refers to how much of that particular thing is present in a given area, right? So outside the cell, we're defining this area, like what is in there. And when we look at the sodiums, I see a lot of these. 1, 2, 3, 4, 5, 6, 7. I could keep counting. There's a lot of sodium out there. All right, so the way we measure that in science is we'll give this kind of complicated looking number. So here's my, oh, it's kind of blocked by this little thing, which is annoying. I'm going to collapse that. All right, so intracellular, so I've got this complicated looking phrase here, and then I've got these complicated looking numbers here. All that's saying, so intracellular, intra means inside, and extracellular means outside. So these terms are just fancy ways of saying that Inside the cell, the sodium is low, right? And we can see that from the picture. There's not very many of those little round balls inside the cell. So the way we describe that in biology is we'll use this crazy looking unit and we'll have a number on it. So this is saying 12 milliequivalents per, so there's a line there, liter. So a concentration is always telling you how much of this substance is in a given amount of space. So the amount of space we're looking at is a liter. If you've ever had like a Nalgene water bottle, that's about a liter of water. So this number is telling you there's 12 milliequivalents, and that unit may not mean anything to you, but you can kind of imagine it as a count of something. So we're sort of going to imagine like this is saying there's 12 of these sodium ions in a liter of water. So we're kind of thinking inside the cell, there's only 12 sodium ions in that amount of water. Now, outside the cell, extracellular sodium, very different situation. You know, we look out here and we see tons of these guys. So not surprisingly, this number is much bigger. So that's a concentration measurement. So we've got 140 milliequivalents of sodium per liter outside of the cell. So we've got this big pileup of sodium outside of the cell. And they're all bopping around and running in their straight lines and crashing into each other. And we know that if we let them be free, you know, if we don't put anything in their way, they're going to do what molecules always do and just spread out and diffuse and end up everywhere. And then eventually they'll be equal in all areas that they're free to move into. But right now, we've got that sodium kind of trapped outside the cell because we haven't opened the channels for it. Now, so potassium, the little diamonds, is in a different situation. We've got a lot of potassium inside the cell. If we look outside the cell, I've got one, looks like two kind of hiding, 3, 4. So very few potassiums outside of the cell, a lot of them inside the cell. So we've got, again, measurements over here. Intracellular potassium, so inside the cell potassium is very high concentration of potassium inside the cell. Extracellular, so outside the cell, potassium concentration is very low. So this is actually reflective of how your cells are most of the time. There's a lot of sodium outside of the cell, very little sodium inside of the cell. There's a lot of potassium inside your cell and very little potassium outside of the cell. As long as the channels are closed, that's just the way it's going to stay. So our potassiums are going to bounce around, they're going to hit each other and knock into each other. They're in motion all the time. You know, they're zooming around. That's our model of diffusion. But they can't get across the membrane. The membrane is a wall to them. Now, if you open a channel for them, things are different, you know, because like, let's say if we open the channel, this guy is bopping along in a straight line. Oh, well, now he can get through this channel and he might kind of bounce his way through, but then he's going to get out. So that's what we see when you have a big pileup of something in one place. If you then let it move, just because these molecules are in constant random motion, they will just randomly spread everywhere. So all you really have to do is get out of their way, and they're just going to spread themselves everywhere. You don't have to do anything. So this is the setup right before the channels open, right? So if we then, oh, and I didn't mention, so I've got oxygens here too, all right? So if we look at oxygen, I don't have listed a concentration for oxygen, but we can see there's only one inside the cell, and there's a whole bunch outside the cell. So our model of diffusion says that if those oxygens are free to do whatever, they're going to spread out until they're equal everywhere. And right now we have kind of a pile up of them outside of the cell. So we know diffusion, our model of diffusion tells us it won't stay that way. If you get out of those oxygens way, they'll spread out and then they'll kind of take up resonance, like this guy may be moving straight in this direction, this guy may be moving in this direction, it'll bounce off, they'll, you know, end up like coming down through here. So they'll spread out everywhere, right? If there's something in their way, they can't do that. So if, like, say the membrane is just a wall to them, and there's no channel for them, then we might expect, well, they're just going to have to stay put. They'll just bounce around where they are. And then if we let them move, they'll spread out everywhere until they get to this equilibrium where they're equal kind of in all areas. So this is our scenario right now, right? So the channels are closed, everything's kind of stuck where it is, just bouncing around in place. And then we're going to open those channels, all right? So here's a scenario, I've cleaned it up a little bit. Now we've got open channels. So sodium and potassium channels are the only channels present in this membrane, all right? So we've left them open. So now these channels are open. I've cleaned up the picture. I no longer am showing you the sodium and potassium, but now I've got these kind of complicated things. This is, I usually, when I look at something like this, I have an initial kind of like, and anxiety response, like, what does that mean? And then once I learn what it means, then I feel pleased with myself because I'm like, ooh, look at me. I understand what that means. So you'll see here, these things are in brackets. So we've got our sodium in brackets here and potassium in brackets, and we've got that on both sides. So again, this is outside the cell. This is outside the cell. This is inside the cell. All right, so these concentration numbers are still telling you the same thing they were before. So if you have a big number for a concentration, it's saying, okay, in this amount of space, there's a lot of these things. So if we have 75 milliequivalents per liter, that's saying, in a simplified term, which I always try to do, and you should try to do too, simple is generally best, we've got 75 sodium ions per every liter of, watery stuff in the cell. So before our previous situation, we had a lot of sodium outside the cell, not very much inside the cell, but the channels were closed and nothing could happen. Then we opened the channels. So now we see outside of the cell is 80 milliequivalents per liter. Inside of the cell is 75. Those numbers are now very close. So if we were to illustrate the situation, okay, so my sodiums are round spheres. All right, so maybe I'm going to throw some sodiums in there. I'm just going to show myself, like, okay, so I did 4 out there, 1, 2, 3, 4, maybe another one there. So that's my visual of what happened here. The sodiums did what I expected. They were stuck on one side of the membrane until the channel opened for them. So this channel is now open. Just by the randomness of diffusion, they just kept bopping around, doing their normal movement thing. And some of them, kind of happen to bob through the channel, and then maybe this one kind of leaves. I mean, they're not directed. They're not trying to get into any particular place. They don't have a brain. They just move in a straight line. They bash into things. And so you give them enough time and they'll spread out everywhere. And so now we've basically reached equilibrium. So this number is almost the same as this number. 75 milliequivalents per liter on one side of the cell membrane, 80 on the other. They're not exactly the same, but we know that's pretty typical because they're always sort of still like drifting back and forth. They don't stay put. So equilibrium doesn't require exactly the same concentration on both sides, but very similar. And we see potassium did something kind of similar. So we've got numbers that are not identical, but they're pretty similar. So if I go back up here, our potassium was, so I had 140 milliequivalents on one side, and then, oh no, potassium, that was sodium. Potassium was 140 on one side, and only 3.5 or 5 on the other side. So now I see those numbers have come much closer together, and that's what we expect, right? So potassium just through its random motion, diffused across the membrane. You give it enough time, it gets to that equilibrium point, and now you have about the same amount of potassium on the outside of the membrane as the inside. So that's what we expect, right? So if something's able to move, and so far we've seen it seems to need a channel to do so. It can't just cross the membrane. The membrane seems to be a wall. That's what we expect to happen. So they've spread out to equilibrium. But if we look at oxygen, so here's a curious thing. In our original scenario, the channels were closed, cell membrane is in place. We had a lot of oxygen piled up outside the cell and very little oxygen inside of the cell. We gave it some time. The only channels in this membrane are sodium channels and potassium channels. And we know that channels are specific. They don't just let any old thing through. They're purpose built. So a potassium channel only lets potassium through. A sodium channel only lets sodium through. I don't see any oxygen channels here. But what I see has happened with oxygen is I've got 1, 2, 3 on the outside, 1, 2, 3 on the inside. So they appear to have done the same thing the sodium and potassium did, which is spread out until they get to equilibrium and they're about equal on both sides of the membrane. And then, they may drift back and forth, but overall you don't see a lot of change there. But what's weird is that oxygen, I don't see any such thing as an oxygen channel in this membrane. And somehow they seem to have managed to diffuse to that equilibrium point where they're equal on both sides of the membrane. So somebody moved. These oxygens somehow crossed that membrane. So what we know is diffusion tells us this universal model of things won't stay in one place. So if you drop some dye into water, all these molecules are in motion. So they're constantly knocking into each other, they're moving in straight lines, and they're hitting each other, they're hitting the walls of the container. So they'll go in a straight line until they hit something, right? And so because they're always doing that, we end up with an equilibrium where everything is spread out. Now, these guys are all still in motion too. So we always have to kind of imagine them like animated in our minds. They don't stop moving. They're always still moving and bouncing and hitting each other. But you won't be able to keep them in this area unless you put a wall around them, right? And if you put a barrier around them, you can keep them crowded together. But if they're free to move, they're just going to spread out everywhere. So we'll end up at this equilibrium, right? I don't know why I choose to write these things when it's such a sloppy thing. But so diffusion is this model where we're like, okay, the molecules are constantly in motion. They move in a straight line until they hit something or something hits them. And because of all this random just kind of bumper cars back and forth, if you give them enough time, they'll spread out to all the areas they can possibly be in, and they'll be about equally spread out everywhere. That's equilibrium. But now we're seeing that sometimes things have barriers and sometimes they don't. So we've got our cell membrane. What we've seen is something like sodium, Na+, sodium ions. It can't cross the membrane on its own, right? If you want to let sodium across a cell membrane, you have to put a channel in the cell membrane and open that channel, let that channel be a passageway, and then sodium will be able to diffuse and it'll cross the membrane and you'll have equal amounts of sodium everywhere in the picture. That's equilibrium. So what we see is that certain things like sodium have that issue. So this little arrow here is saying can't cross without a channel. So to those things, with this kind of bouncing off arrow, the membrane is a barrier. And unless they have a channel to go through, they won't diffuse across that membrane. They'll keep bopping around on one side of the membrane, crashing into each other, crashing in the membrane, hitting whatever else is there, but they can't get across the membrane without a channel. That's not the case for everything. So what we saw with oxygen was there was no such thing as an oxygen channel in the picture, but somehow you just let time pass and oxygen went from really piled up on one side of the membrane to equilibrium on both sides of the membrane. It was equally spread out everywhere. So oxygen was behaving like the cell membrane was not a barrier to it. There was no channel for it, and yet it was able to do this. It was able to just cross the membrane directly. So there are categories of molecules that behave in these different ways. All these guys over here. So amino acids, remember those? They're the building blocks of proteins. If you break a protein into its component bits, you get a bunch of amino acids. If you have those amino acids in your body, they can't cross your cell membrane unless there's a channel for them. That's also true for the glucose that you get from your food, that your body needs to survive. Glucose can't cross the membrane without a channel. If there's a channel for glucose, if there's a tunnel for it, it will diffuse across the way we expect. But without a channel, it's effectively trapped on one side of the membrane. Other things just don't see the membrane as a barrier at all. Carbon dioxide, whatever, it can go through. N2, nitrogen, so this nitrogen gas, it can go through. Glycerol is another thing that is part of your diet that you use. It doesn't have a problem crossing a membrane. Ethanol, we've seen that, it can cross. So there's certain kinds of molecules that the membrane is not a barrier to. It's like it's not even there. They just drift right across it. No big deal. There's other things that behave totally differently. They come up against a membrane and they bounce right off it, like it's a brick wall to them. And then you've got some, like water, H2O. I've got this little arrow here that's really trying to show you that oxygen kind of behaves somewhere in the middle. When an oxygen kind of drifts up to a membrane, it can cross, but it'll take kind of this like slow, like, okay. So water behaves something in the middle. These guys all breeze right across it. It's like the membrane doesn't even exist to them. They just drift across it. If that's where diffusion leads them, if glycerol happens to be traveling in a straight path, the membrane is not going to stop it. If this oxygen kind of bounces off the side here and then comes back over here, it's just going to pass right through that membrane like it's not there. If this glucose is traveling in a straight line, it bounces off this surface and it bounces back, it's going to hit the membrane and bounce off again. So that's pretty weird. This is like the cell membrane is acting sort of like some sort of sci-fi force field that's there for certain things but not for others. And then water hits it and doesn't bounce off, but like it gets slowed down by the membrane. It's like, like I'm going in my straight line, but it's difficult. So we've got things that easily cross the membrane like it's not even there, other things that bounce off it like it's a brick wall, and then things like water that can pass it, but will get slowed down. It's kind of like it hit like a mud pit and is slogging through, but it can get through. So we have to think like, and you can, this is one of these things where I'm not going to tell you exactly what to look at for this, but you can look at these different molecules and just start to make some predictions. Like, what is it that seems to make certain things see the membrane as a force field, can't cross, just bounce right off. Other things sail right through with no problem, and other things can get through but take this, I'm really enjoying making this squiggly, it's very satisfying. Other things kind of struggle across the membrane, can cross it but with difficulty. So you may come up, like just scanning over this, you have a perfectly good brain, see if you notice any patterns or anything that seems to be consistent or anything that's sometimes consistent about what things easily cross the cell membrane, what things bounce off like it's a brick wall, and what things kind of struggle. And that might not be obvious to you. And you're welcome to look anything up. If you're just like, well, I think it might be this, but let me go find out. That's the key to education, because I'm not always going to be here in your life to tell you what to go look up. If you're curious about it, look it up. We'll get some understanding together, but there's a lot of stuff that you can just look up for your own sake. So look at these images a little bit, try and think in your own mind what seems to determine which things bounce off the membrane and can't cross it unless they're given a channel, what things sail across it like it's not even there, and what things kind of struggle. So what we're going to move into is some discussion of that. What is it that drives that? And we're going to have, so we've got diffusion as one of our mental models that we're building. We kind of can picture how molecules move, how they get to equilibrium, why they change direction, if they change direction. We need an additional governing principle that's going to carry us through the entire rest of the class, all of your chemistry education, all of your biology education. And that is this rule, which you already know about. You already know that positive and negative things attract. The reason you know that is because if you take magnets, right? So magnets don't always look like this, but magnets have two ends, right? So they have a positive end and a negative end. If you take another magnet and you bring it over here, and in this case, we've got a positive and a negative. I'm going to change my color. So if you do that, what you're going to see, I did not change my color. Why does this always happen to me? Okay. All right. So this is the ultimate governing rule, really, that runs all of chemistry. If you see a negative sign and a positive sign, they're going to love each other. So if you have a positive end of one magnet and negative end of another magnet, they're going to snap together. That attraction is all positively charged things will be attracted to all negatively charged things and vice versa. So opposites attract, all right? So this is our governing principle. Opposites attract. All right, so then if we take two magnets, and instead of that, we say, okay, I'm going to face the negative end of this magnet and the negative end of that magnet, what we're going to get is, this is like counterintuitive. Good Lord. Just trying to do this cool. All right, so this is my universal symbol for do not get along. Okay. This is very elaborate. All right, so like charges repel. So they hate each other. So if you try to put two negative charges near each other, they're going to push apart. They try to escape from each other. The closer you try to force them to be, the more they hate each other and will force themselves apart again. The same thing is true for two positives, right? So two positives, also skull and bones. I'm not going to do as good a job. Kind of can't help myself finishing. Well, that looks really, really bad. All right, anyway, that's true of this case too. You try to bring two positive charges together, they will try to get away from each other. They will force each other apart. The more you try to push them together, if you take two magnets, the wrong sides of two magnets, you can try it with refrigerator magnets, they will keep sliding off of each other. And when you try to actually force them to touch, they just will refuse. They will just keep launching off of each other. So that is like charges repel. Two positives basically hate each other. You can think of it as hatred. They're going to try really hard to get away from each other. The closer you try to make them, the more they will force each other away. Positives and negatives are always going to be attracted to each other. And if you try to bring them closer together, you're going to feel that attraction even more. So both the attraction and the repulsion will get stronger the closer they get to each other. And so same charges repel and opposite charges will attract. Now, the other thing is if you've got a positive and a negative, I'm going to switch to this, hopefully. Okay, so they'll like each other, right? But the other kind of rule here, so positives and negatives will always like each other. They'll always attract. The bigger the positive charge and the bigger the negative charge, the bigger the attraction. And you can think of that like magnets too. If you have really little refrigerator magnets, yeah, you can feel the attraction and repulsion as you play around with them. But if you have big industrial magnets, those can get scary, actually. Like if you put your hand between industrial magnets that are like positively and negatively charged, they'll break your bones as they snap together. We've actually got cases in medicine where children and animals will swallow magnets, and the magnets will pull towards each other in the intestinal tract. And as they snap together, they can actually burst through the wall of the intestine. Then you need emergency surgery. So this is not a force to be trifled with, and this is a governing principle. Positives and negatives will always pull towards each other. The bigger the charges, if you have strong charges, strong positive, strong negative, you're going to have more of a force pulling them together. If you have big positive charges against other big positive charges, you're going to have a strong repulsion. They're going to push apart really hard. So these are the basic laws that I want you to carry forward into the next assignment I have you guys doing. If you can keep these mental models all together in your head, right? So our diffusion model is that molecules are always in motion. They travel in a straight line until they hit something and bounce off. They're just kind of indifferent to each other. But now we're going to get a little more complicated, because if you have particles that have positive charges and other ones that have negative charges, they're not indifferent to each other anymore. They won't just kind of ignore each other until they crash into each other. They actually will respond to each other, because positives and negatives are going to pull towards each other. Two positives are going to push away from each other. Two negatives are going to push away from each other. And now we're going to see how that, so what we're talking about here is the electrostatic force. So that electrostatic force, you can think of it as a magnetism. And just like magnets can be drawn towards each other if you have them facing the right way, or they can push apart if you have them facing the wrong way, that is how everything that we see for the rest of the semester is going to be governed. And that's what runs all of chemistry. It is as simple as that. Look for positive charges, look for negative charges, make your prediction about how they'll behave based on whether they will be attracted or repelled from each other, and you have effectively built a mental model that will carry you all the way through an entire science education. I am not joking. It is that simple. We're just going to add nuances to it and make it even cooler, but that mental model is going to get you